Okay, part one, understanding the cell cycle. We'll start out with a brief description of what happens during the cell cycle. So a cell has a cell wall, it has a nucleus, it has DNA within that nucleus. And so this cell has a process called the cell cycle, and the idea behind the cell cycle is just simply that the cell can eventually duplicate into two cells. It can divide and replicate into two cells. And so the first thing to understand is just the different phases of the cell cycle. So G0, this is a phase when the cells are not dividing, and typically they're often also not growing. So, if you think about many cells that have differentiated into skin cells, into neurons, into heart cells, most of those have reached an endpoint, and they are no longer able to divide to form new cells. They perform the functions of a neuron, they perform the functions of a skin cell, etc., but they are unable to actually divide any further. So division occurs using specified cells, typically called stem cells, for example. And so it's only a select number of cells that are able to go through this whole process. So the other cells end up differentiated, and they are typically stuck in this G0 phase. So if you're a stem cell, though, you can actually continue to go through the cell cycle and enter these additional phases. So part of what you need to do in order to divide is grow, and so typically the G indicates some sort of growth. And so the cell that is going through the cell cycle gets a little bit bigger. So it produces more proteins, it produces more cell wall, it produces more ribosomes, it produces more organelles. Anything that a cell needs, you're going to have to double the content before you divide. And so the first thing that happens, this occurs during G1. Now, one of the things you also need in order to replicate a whole cell is you have to duplicate the entire copy of DNA. So each cell must have a copy of the DNA instructions that allow it to know, am I a human cell, am I a dog cell, etc. And so S phase is the phase where DNA replication occurs. S, synthesis, that is when DNA is replicated. So every single DNA molecule that's within the cell needs to be replicated. After that, there's G2 phase. So G2 phase is when the cell continues to grow, new ribosomes, new cell walls, etc. are all formed. And then once the cell has actually grown to the point where it has enough material to make two cells, then it is ready to actually divide. And so one of the most important steps of division is actually ensuring that each cell receives an appropriate content of DNA. So the entire genome must be passed on to each cell. And so this is why we talk about mitosis and meiosis, is that these are the phases that ensure that DNA is properly segregated to each one of the cells. So one thing to remember is that mitosis and meiosis are just a small part of the total cell cycle. So when you think about the total cell cycle, most of the time the cell spends is actually growing, replicating DNA, growing, and a very small fraction of the time is actually spent in mitosis or meiosis. So mitosis and meiosis are just a subsection of the cell cycle. Interphase refers to the phases of the cell cycle that don't include mitosis or meiosis. So interphase is all this part of the circle with the exception of mitosis and meiosis. So interphase is a good phrase to remember. It's a phrase. It's a term that you'll be expected to know, for example, for the test. Mitosis is further divided into multiple phases. So prophase, prometaphase, metaphase, anaphase, and telophase. And we'll go over the exact series of events that occur during those stages. So karyotype analysis. A karyotype is a photograph of chromosomes in a cell. So they're typically taken from a blood cell, a fetal skin cell, or a bone marrow cell. So these are cells that are actively dividing and in mitosis. So one of the things about a karyotype is that you cannot take a karyotype of a cell that's in S phase or any sort of interphase. You have to actually identify a cell that is in meiosis or mitosis in order to perform a karyotype. And the reason for that is simple. It's that chromosomes only form during mitosis or meiosis. And so if you were actually an experimenter that was trying to perform a karyotype, one of the things you would need to do is identify cells that are undergoing a large amount of cell division. And so this is why they can be taken from blood cells, fetal skin cells, or bone marrow cells, because these are cells that are very actively dividing and in mitosis. So the goal is to take a picture of all of the different chromosomes within a cell. So you have to identify dividing cells. So if you take a sample of bone marrow cells, some of those cells are dividing, some of those cells are not dividing. So one of the things you have to do is identify the cells that are dividing. You have to release the contents of the cells through lysis. So lysis is a term that's used to indicate that. You have treated typically with either a chemical, or you treated the cells with some sort of physical force in order to break apart the cell membrane to release the contents of the cell. Then you stain the chromosomes using a dye, so you want to be able to see the chromosomes, and then you simply sort the chromosomes by length and homology. And so the released chromosomes might initially look something like this, where they're kind of all over the place, and then the experimenter will actually identify chromosomes that share homology, and then sort them together and order them. So humans, you should all know, have 23 chromosomes. So 23andMe, for example, is a company that you can use to get information about your own DNA. And the name comes from the fact that we each have 23 unique chromosomes. We are diploid, humans are diploid, and so you'll notice that each chromosome has 2 copies. So there's 2 copies of chromosome 1, 2 copies of chromosome 2, etc., etc. And so if you go through, each one of the chromosomes has 2 different copies. So using that dye, using the staining from the dye, you'll notice that each chromosome stains a different amount at different positions. And so that's one of the keys to the dye. So the dye not only allows you to see the DNA, but it also allows you to distinguish homologous chromosomes, because the homologous chromosomes will die in the same places. So part of the way that you determine this is chromosome 1 is the length. So chromosome 1 is the longest chromosome. And then also you notice that it's much brighter at the tip here, and et cetera, et cetera. So these patterns are also what the experimenter uses in order to sort the chromosomes together. So you'll notice that there are 23 chromosomes, and the one exception of having 2 copies of each is the sex chromosomes. So there are 22 autosomes and 1 sex chromosome. And so the sex chromosome depends upon your sex, whether or not you have 2 copies of the X chromosome. So your 2 copies of the XX would be the female sex, and then 2 copies of 1 copy of X and 1 copy of Y would be the male sex. And so that is the one exception to each individual having 2 copies of each chromosome. So females would have 2 copies of the 23 chromosomes, males have 2 copies of 22 of them, and then they have 1 X and 1 Y, until you see that there's a huge size difference between the X chromosome and the Y chromosome. The Y chromosome is very small, it does not encode many genes. The X chromosome is a typical sized chromosome. So this karyotype is one of the first things that was used in order to figure out kind of the rules of inheritance and the rules of DNA. And so these chromosomes are something that would appear within a karyotype. So chromosomes, we kind of have a loose definition. We kind of refer to chromosomes in kind of ubiquitous ways, not always appropriate. So a chromosome, for the purpose of this class, the way we refer to a chromosome are these physical structures that form. And so it's something that you would be able to visualize in a karyotype, because the DNA condenses and forms in these very specific type structures. So chromosomes were discovered in 1843. So, a long time ago, people were able to actually see these structures And initially all they had to use was light microscopy in certain cells. So, before they discovered DNA dyes that can actually stain those, these chromosomes were large enough that they could be seen in actual light microscopy. So chromosomes are only seen at certain times in the cell cycle, and they're only seen after DNA replication has occurred. So if you grab a cell, if you grab a normal skin cell and you looked within that cell, you would not see these chromosomes because most cells do not organize them into these chromosome structures, or most cells don't organize their DNA into these chromosome structures. So chromosomes are highly compacted forms of DNA. The DNA gets more and more condensed. Studies showed that DNA is organized into multiple chromosomes. Some chunks of the DNA were very similar. Humans and most animals have 2 copies of each chromosome. And so this is where the name diploid comes from. We have 2 homologous copies of each chromosome. So the diploid number is equal to 2 times n. So if I say an organism has a diploid number of 86, I'm giving you a lot of information. I'm telling you, 1, that the animal is diploid, so only diploid animals can have a diploid number. 2, I'm telling you the total number of unique chromosomes. So the diploid number is equal to 2 times n, where n is the number of unique chromosomes. So humans have 23 unique chromosomes, and our diploid number is 46. And our diploid number, of course, is also the total number of chromosomes you would see in a karyotype. So if we counted 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, we would count 46 unique chromosomes within this karyotype. So chromosomes, what are they made of? They're composed of DNA and protein. So the orange kind of indicates the condensed DNA molecule. They are held together. There are actually 2 DNA molecules within this chromosome. And as a result, we have this other term to indicate that there's actually 2 chromatids within each chromosome. So there's actually 2 DNA molecules. So this right here is 1 DNA molecule. This right here is another DNA molecule. And these are condensed down. So there's all sorts of proteins that are within this that hold all of that DNA in that condensed manner. All of this right here is similarly. And so each one of these is called a sister chromatid. And then these are also held together. So the sister chromatids are held together by an important protein called cohesin. And so this is kind of the glue that holds these things together. You also have a kinetochore. So the kinetochore becomes very important for the purpose of dividing DNA into the different cells. So this is a location where the microtubules can actually attach onto, and eventually these 2 sister chromatids are going to be separated and moved to different directions. And then there's also some important proteins, shugashin. I don't actually know how to say that. You won't be expected to know that. But that is actually an important protein that's degraded that allows for separation to occur. So the centromere, that's an important term. Centromere is the region where the kinetochore forms, and this is important for dividing chromosomes. So this is a process. So maybe you sort of know what's in a chromosome, but one of the important things that you're expected to know for this class is actually a much more detailed description. So you need to know that there's 2 DNA molecules within a chromosome. These DNA molecules are exact copies of each other, and the reason they're held together is that they're actually created by the process of DNA replication. So if we go all the way back to the cell cycle during the S phase, each DNA molecule is replicated. So you have a double-stranded DNA molecule during synthesis. This forms a second exact copy. So this is a second double-stranded piece of DNA that's an exact copy. And so this is what eventually becomes the chromosome. So each one of these DNA molecules is held together, and each one forms a different sister chromatid. So chromosomes are composed of DNA and proteins. Centromeres are regions where the spindle fibers attach. So these are what causes this characteristic X shape. And the position of this X shape is determined by where the centromere is within the chromosome. Telomeres are the ends of the chromosomes. So telomeres are the end of the linear molecules. And chromosomes are often named based on the position of their centromere. So the centromere can be in the middle, it can be between the middle and the end, it can be close to the end, and it can be right at the end. So you could see something like this in a karyotype, or this is kind of another type of image of a chromosome. And so where exactly that pinching point is, if it's in the center, then it is a metacentric chromosome. If it's closer to one arm, it's submetacentric. If it's almost at the end, it's acrocentric. And then if it's all the way at the end, it's telocentric. So telomeres, telocentric means it's almost at the end. This also allows us to add another naming convention in that we have two arms of the chromosome. So there's the P arm. So the P arm is always the shorter, and then the Q arm is always the longer. So as you get more, if you become a human geneticist, or if you're really into human health, you might get into human genes, and human genes often have very specific positions. And the position is often named with the number of the chromosome and then whether or not it's on the P or the Q arm. So those P's and the Q's are useful to know because they become important for naming conventions of locations of genes and other loci that might be of interest. So if you're interested in GWAS, if you've heard of anything like that, then those are also important. So again, each chromosome contains two sister chromatids, and sister chromatids are created by DNA replication. So human chromosomes, there are 23 unique chromosomes, 22 autosomes, and then the sex chromosomes, and each one has two homologous chromosomes per unique chromosome, so their diploid number is 46. And so this just shows kind of like the distribution of both the sizes of chromosomes for humans. I just pulled this from Wikipedia. So the largest, so these are almost all named in order of size, with the exception of 21 and 22 are reversed. But you'll see that the largest chromosome is chromosome 1, the second is chromosome 2, etc. So there's 247 base pairs of DNA in chromosome 1. And then the smallest is chromosome 21, which has 46 million base pairs of DNA. The X and the Y, you'll notice, have very different sizes. So the X chromosome is 154 megabases, the Y chromosome is 57 megabases. So the Y chromosome is actually not the smallest chromosome. 21 and 22 are actually the smallest. And then if you look by the number of genes, so there's not a exact linear monotonic effect. So the larger chromosomes tend to have more genes, but you'll also see that there's a lot of exceptions to that rule. So for example, chromosome 19 has 1,500 genes, which would place it as number 2 in the order. So number of genes don't equal the number of base pairs. So their chromosomes are typically named by the base pair's size, not the gene content. And there's a huge range of distribution of genes on the different chromosomes. So chromosome organization is unique to its own species. So we have a diploid number of 46, but if you have any sort of pets, if you have a dog or a cat, they would have a different diploid number. So it's not really understood in detail why this happens, but in the course of evolution, there's often fusing and dividing of chromosomes that occurs so that each organism might have a different number. So these are examples of 3 different species. All of these species are diploid, but one has a diploid number of 26, 32, and 66. And if you did a karyotype, you'd be able to recognize that because you would see that each chromosome has another homologous chromosome, so it's diploid, and then the number of chromosomes would be different. And this is a table that kind of shows even more. So kingfisher is one of the animals that has the most chromosomes. It has a diploid number of 132. And the fruit fly, Drosophila, it's a very common research organism. That is one of the organisms that has one of the fewest number of chromosomes. So its diploid number is 8. So homologous chromosomes. So remember, sister chromatids are exact matches to each other because those are created by DNA replication. Homologous chromosomes are similar to each other, but they're not identical. So what is a homologous chromosome? So you receive a chromosome, a copy of chromosome 1 from your father and 1 from your mother. So the reason you're diploid is that you inherit your DNA from 2 parents. You don't have a single parent, you have 2 parents. And so your father provides you with 1 copy of a chromosome 1, and your mother provides you another copy. So these chromosomes are very similar to each other. So they carry the same set of genes. So if you have hemoglobin gene on 1, if you have myoglobin gene on chromosome 1, you would receive a copy from your father and a copy of your mother. However, there could be slight variations within the gene, and these are called alleles. So if you look at the base pair levels, if you have A, C, C, G, G, T from your mother, most of that would be the same, but there might be an exception, and you might have a G instead of a C at one of the positions. So this C, this should be right here. This base pair is a C in one chromosome and a G in another chromosome. So there's subtle differences in the nucleotide sequence, and that could lead to either subtle or profound differences in the function of the gene. So this can be a cause of disease. So many diseases are caused by loss of functions in a gene, so the gene no longer works. They could have no effect, so there's also mutations that modify the nucleotide sequence, but they don't actually affect the function of the gene. So when we talk about having homologous chromosomes, it's important to remember that we have 2 copies of each gene, but there's subtle differences between them. And on average, there's something like 1 out of every 1,000 base pairs is different. So if you looked at your 2 copies, and you read this DNA, you would have to come to about 1,000 base pairs before you found a difference between these. But overall, because there's so much DNA within the cell, there's actually a large amount of differences. So, we go back to this table right here. So that would mean, on average, there's 247,000 differences between your homologous chromosome 1. So you got 1 from your mother, 1 from your father, and if you counted how many differences there are, on average, there's approximately 247,000 differences just on chromosome 1. So there's actually a huge amount of differences at the DNA level between these homologous chromosomes. So typically, homologous chromosomes are identified by size, so they have the same overall length. Centromere position, so of course the centromeres are going to be in the same place where that pinching occurs, and then the banding pattern for DNA dyes. So there's multiple different DNA dyes that you can use, and you can choose one that make it obvious that the banding pattern is the same. So of course, bacteria and viruses, this is a rule for eukaryotic individuals. So of course, bacteria and viruses, chromosomes are excluded from this. There are also organisms, and in general, it is rare. Almost all organisms are either haploid or diploid, but there are organisms that can also have more than 2 copies of each chromosome. So a triploid organism is something that has 3 copies of each chromosome. So 3 copies of chromosome 1, 2, 3, etc. And a tetraploid organism has 4 copies of each chromosome. So again, you can readily identify these in the karyotype because you would see 4 exact copies of chromosomes that have the same size, that have the same centromere position, and have the same DNA binding pattern. Genome. So what is a genome? So I'm sure you have heard the term genome. So the human genome was sequenced back in 2002. You've heard that we have genome files for all sorts of organisms. So what exactly is meant by the term genome? So again, in this class, you need to get specific about your knowledge of these terms. So genome is the DNA content of a haploid cell. So what is the DNA in a sperm cell or an oocyte? So in these gametes, each individual cell is haploid. So in a sperm cell and an egg cell, these are special cells. So they only have 23 chromosomes, because when they fuse together, they come together to produce a new individual that has 46 chromosomes. So a sperm cell or an oocyte only has 1 copy of chromosome 1, 1 copy of chromosome 2, etc., etc. So these are haploid cells. They have 1 copy of each chromosome. So the genome, when we refer to the human genome, what we're referring to is the size of the DNA within a sperm cell or an oocyte cell. And so if we had a file, for example, if we had a file that listed the genome, if you downloaded the human genome, there would be chromosome 1, and then there would be, 257 million bases that would be listed to list the content of chromosome 1. And then you'd have chromosome 2, and then you would list every single base pair of chromosome 2. So I might give you the genome size, and the genome size would just be this right here. So the human genome size is 3 gigabases. And so you can get this just by adding up all of these numbers. So if we add up all of these numbers, you would end up with a total of 3 gigabases of DNA, of unique sequence. But if I asked you how much DNA was in a diploid cell, because diploid cells have 2 copies of each homologous chromosome, there would be more base pairs of DNA. And then you also have to remember, has DNA replicated or not, because that will also influence how much DNA is within a cell. So the purpose of your activity is to kind of go over that in various details. So just the end of part 1, so the genome, the genetic material of an organism. The genome is the sequence of nucleotides. It can be contained in a text file. It is the size of a haploid genome. A DNA molecule, 2 long chains of nucleotides twisted in a double helix. Each cell contains on the order of 100 DNA molecules. A chromosome is the organized structure of DNA. It can contain multiple DNA molecules. It contains proteins, and it actually also contains RNA. A chromosome can only be seen during mitosis or meiosis. A chromatid is 1 copy of a duplicated chromosome joined at the centromere. Homologous chromosomes are a maternal and paternal complement of the same chromosome. They pair up with each other during meiosis. And a sister chromatome refers to 2 identical copies formed by the replication of a single chromosome. So again, it's important that you understand all of these terms well, what exactly is meant by them, and actually just understanding this terminology is half of the battle for understanding this material from this chapter.